Our last speaker is Todd Schoborg from NIH, and he's going to be telling us about mechanisms of centrosome inheritance.
Okay, so thank you all for coming. The story I'm going to tell you about today is some work that I've been doing on two proteins, abnormal spindle and Calmodulin. And this originally started off as a cell biology story, but has since somehow transitioned into a neurodevelopment story. So I'm a postdoc in Nasser's lab, and the Rusan lab at its heart is a spindle lab. But essentially we're broken up into two camps. There's some people that are interested in the structure, function, and regulation of the centrosome, which functions as a primary microtubule organizing center in animal cells. Then there's the other camp, which I sort of belong to, which is just simply more interested in the structure and dynamics of the spindle itself. And so this is just a movie right here that's showing you the formation of the spindle. And so you'll notice a nice bipolar array of microtubules. And the cell's going to sit here and think about things for a while to make sure all checkpoints are ensured before it goes ahead and executes its most critical task, which is ensuring proper chromosome segregation. And we know from numerous genome-wide screens that there are hundreds of genes that are responsible for controlling this structure. And we know this because of the diverse range of phenotypes that we see when these genes are mutated. But the one common thread among all of these phenotypes is that chromosome segregation is likely to be impaired, right? And so when you have defects in chromosome segregation, this is going to make cells unhappy. And when your cells are unhappy, this is going to make tissues unhappy. And when your tissues are unhappy, under the right or perhaps the wrong circumstances, this is going to lead to human disease. And so this is just a schematic of the spindle itself that I want to use to point out a few key principles. And the first is that this microtubule-based machine consists of three microtubule populations. So we have the kinetochore microtubules. These connect directly to the chromosomes. We have the astral microtubules, which radiate out from the centrosome itself. And then we also have the spindle microtubules located in the lattice here that help give this structure rigidity or support. The other thing I want you to notice is that these microtubules have a very defined polarity within these structures, with the plus ends oriented towards the chromosomes and the minus ends coalescing in a region which we call the spindle pole. And you'll notice that this coalescence gives this structure its diamond or American football shape. And we know that this structure doesn't just function alone in isolation. There are also a number of other proteins that are involved in regulating this structure. And we call these microtubule-associated proteins, or MAPs. And these do everything from helping to stabilize kinetochores, to assisting in microtubule sliding, to the aforementioned pole focusing. And so the story I'm going to tell you about today is revolving some work that I've done with pole focusing, particularly on this protein called abnormal spindle, or Asp. And so most of what we know about Asp comes from the work of David Glover's lab, and they were able to show biochemically that this rather large protein contains an N-terminal binding domain located here. But we noticed that the C-terminus of this protein contained a number of IQ motifs, which are known to bind to the calcium-sensing protein Calmodulin. And so what we hypothesized was that it was this interaction between CAM and Asp that might be important for pole focusing in the fly. And so we set out first to verify this genetically, and we did this using an RNAi-based approach in which we depleted S2 cells with either Asp Cam or a combination of both. And I want to point your attention to two phenotypes here. The first, you'll notice, is this loss of spindle pole focusing. And then you'll also notice that these centrosomes have completely detached from the spindle pole and just sort of randomly floating around the cell. And importantly, we see an identical phenotype when we just deplete CAM alone. And then, perhaps maybe more importantly, we see a no more severe phenotype when we deplete both simultaneously. And so this tells us that these two proteins likely function as part of a complex. And in data that I'm not showing you here today, we do have yeast-2 hybrid analysis that Asp and CaM do interact with each other directly. And so in addition to that, we are also able to use live imaging to reveal some pretty interesting dynamics for these two proteins. So this is an S2 cell that's in metaphase. It's expressing both tag versions of CAM and Asp. And as I play this movie, what you'll notice is that in addition to the very strong pole localization for both proteins, you'll also see these foci that appear to be originating within the lattice itself and then streaming back towards the spindle pole. And so we quantified the rate at which these foci are moved towards the spindle pole, and given the similarity in these velocities to the rate at which tubulin fluxes through the spindle, and this had been previously calculated elsewhere, we favor a model in which these two proteins are physically bound to the wall of the microtubules and are being transported back to the spindle pole via microtubule flux. And so based on this data and a lot of other data, which I don't have time to show you today, we formulated the following model for how Asp and CaM might be cooperating to cross-link microtubules within the spindle. And we envisioned two possible scenarios. The first, in which CaM might be able to help mediate dimerization between two microtubule-bound Asp molecules. Alternatively, CaM might be able to favor some type of conformation in a single Asp molecule that allows it to bind to two microtubules simultaneously. But I think the most important implication from this work is that we were finally able to ascribe a function to the localization phenotype for CaM to the spindle pole. And this is a phenotype that had been known for well over 35 years. We just had no idea what it was doing there. And importantly, we were able to show that the spindle pole localization for CaM is solely dependent on the single protein, which is Asp. So I'm going to switch gears now and talk about the developmental consequences of this work. And what I haven't told you yet about Asp is that Asp is a key determinant of brain size. So there is a vertebrate ortholog of this gene, and it's called, in humans, and it's called abnormal spindle-like microcephaly associated, or Asp, or Asp-M. And the reason it's called that is because it's the most commonly mutated gene in patients who have microcephaly. And so I'm just showing you here a domain representation of where all of these human mutations have been mapped with respect to the protein, and you notice they're distributed throughout. And microcephaly, of course, was introduced in the last talk, and all of us are familiar with it given the current Zika virus outbreak. And so we thought, okay, can we use our favorite model organism, the fruit fly, to begin to try to understand the cell biology underlying this disease? And the reason that we can do this is because the microcephaly phenotypes that we see in humans and other vertebrates are recapitulated in the fly. And this was first shown by Renata Basto's lab a few years ago, and you can see that here both in third instar larval brains and in adult heads, the Asp mutant is significantly smaller than their wild-type counterparts. And so to begin probing this connection with microcephaly, I generated some new tools of my own, and I took advantage of CRISPR technology in order to generate a clean null for Asp. And I did this essentially by wiping out the 5' end of this gene, generating this deletion. And this completely obliterated Asp mRNA expression as shown here. And then most importantly, this also recapitulated our microcephaly phenotype. And so to begin trying to understand the cell biology connection here, I took advantage of another tool, which were flies that were expressing a GFP tag version of calmodulin. And what this allowed me to do is to go into the brain and focus particularly within the neural stem cells, which we call neuroblasts. And this made logical sense because these stem cells give rise to all the other cells in the brain. And so this is live imaging within the intact brain here. So this is a neuroblast that's in metaphase expressing CAM GFP. And what you'll notice is that this concerted streaming movement that we see during metaphase back towards the spindle pole is also conserved in this tissue type, in this cell and in this tissue type as well. But the really cool stuff didn't really start to come out until we started looking specifically at the microtubules themselves, and particularly at the spindle. And so what I'm showing you here, this is more live imaging within the intact brain, and this is labeled with a tubulin GFP. And so this is what a normal division looks like. And so there's a couple things that I want to point out here that I want you to focus on. So first, you'll notice that the centrosomes assist in the nucleation of this structure, and they remain embedded in the spindle poles. And you'll also notice that the spindle has a nice diamond shape and it's straight, and when the cell goes ahead and divides, the centrosomes are partitioned to each daughter cell accordingly. But this is what we saw in the mutant, the Asp mutant neuroblast. So what you'll notice is that as soon as the spindle starts to nucleate, the centrosomes pop off of the end of from the spindle poles, and the spindle does this weird rotation thing. It becomes bent and elongates slightly along the cortex before it will go ahead and divide normally. But what you'll also notice is that these centrosomes, both of these centrosomes, end up being inherited by the same cell. And so based on these phenotypes, we formulated a very simple hypothesis. So we know we have spindle defects. This is going to lead to altered cell division. We're probably going to get some apoptosis as a result of that, and fewer neurons equals a smaller brain. So while this is very simplistic in nature, it's intuitive nonetheless. But what I can tell you, to make a long story short, is that this isn't the case. So the spindle defects that we observed do not correlate with the microcephaly phenotype. And so we did this using a genetic rescue approach in which we made 3 transgene rescue fragments for Asp. We had a full-length version, a C-terminal truncation, and an N-terminal truncation. And what you'll notice is that only the full-length transgene could rescue the spindle defects that we observed in the mutant background. Neither the C or the N-terminal fragment could. But the real kicker came when we started looking a little bit further downstream developmentally. And what we found is that the full-length version could completely rescue the microcephaly phenotype that we observed. The C-terminal fragment could not, so these were similar in size to the mutant alone. But the kicker came from the N-terminal fragment, because despite the fact showing all of these very severe spindle defects, we got a completely normal-sized brain. And so this told us 3 things. It told us, 1, that the spindle defects that we see in the Asp mutant aren't necessarily the main causative agent behind microcephaly in the Asp mutant. It also told us that the Asp Cam interaction is completely dispensable for proper brain size, and that there's something else going on in the N-terminus of this protein that alone is sufficient to determine proper head and brain size. And so what I am doing now currently is going through this N-terminus with a fine-tooth comb, trying to find the smallest domain that I can that will rescue the microcephaly phenotype. And I'm also looking for other cell biology phenotypes that might point towards a mechanism by which Asp is able to determine proper brain size. And so with that, I will thank you for your attention, and I would be happy to take any questions that you might have. Thank you.
All right, we have time for questions.
Yes.
Hi, you showed that the spindle defects don't seem to correlate with microcephaly. Do the centrosomal inheritance phenotypes?
No, so they don't either. Yeah, in either case, we see centrosomes just being completely inherited at random, even in that N-terminal fragment that rescues brain size.
What is the in vivo phenotype of calmodulin? Do you actually see the the detachment of the centrosome and the dissegregation of the centrosome? And the second question is, can you delineate what happens to the cell that inherits the 2 centrosomes versus the cell that actually doesn't have a centrosome?
Sure. So to answer your first question, we haven't been able to look at any type of CAM mutant phenotype in the animal because it's lethal. And, CAM does so many things in the cell, particularly during interphase, that's just going to be a lethal phenotype, so we haven't been able to parse that out. Your second question, have we been able to follow the behavior of the 2 centrosomes after they've been inherited? We haven't tried to do that, but I have observed like neuroblasts that will have 3 or 4 centrosomes in them that are still going on and dividing normally.
In your rescue,
Yes.
especially the N-terminal one, did you look at the numbers of neuroblasts in the larval brain?
Yes, so I didn't look specifically at the N-terminal fragment, but just counting neuroblasts in the Asp null mutant that I made, in larval, third instar larval brains, there's not a difference in neuroblast number.
How about apoptosis? Did you look at that?
So I haven't looked at apoptosis yet. That's something that I'm currently doing.
Hello. So I just have a curiosity.
Yes.
In your movies of CAM, you could see really nice localization after the chromosomes have been segregated in the spindle midzone. Does Asp also localize there, or it's something?
So, yeah. So both CAM and Asp will go to the central spindle region. Sort of the peripheral association that you saw with like the astral microtubules. Asp also shows that same localization, and we have no idea what it's doing there.
Yeah, that's very interesting.
Yes.
All right, if there are no more questions, let's thank Todd and all of our speakers for a fantastic session.